Hello. Welcome to the next in the series of review clips designed to assist you in preparing for your Biology 301 public examination. In this clip, we'll be taking a look at genetic engineering. By definition of genetic engineering is the manipulation of an organism's genetic material to modify the proteins that it produces. There are a number of ways this can occur, and there are several steps that are involved in manipulating or even modifying the DNA of an organism. We'll go through the steps or techniques on the next few slides. One of the first methods used is the use of what are known as restriction enzymes. These restriction enzymes are, as their name suggests, enzymes such as restriction endonuclease. These enzymes are used to cut DNA into short fragments. And each fragment of DNA will have a sticky end. These will be short segments of unpaired nucleotides on each end of a fragment. The restriction enzymes cut along specific base sequences. So when the DNA fragment is cut, the sticky ends will be complementary to a corresponding set of nucleotides. By way of illustration, if you take in E. coli, one particular restriction enzyme is designed such that it will cut along this base sequence that you see along here. This will always be the case. So when you cut an adjacent set of DNA and you create a sticky end with this pairing, you can then cut the host DNA that you want to insert in the same place. By so doing, you are able to create what are known as recombinant DNA. You are thinking then, once you use restriction endonuclease to cut, you can create a new segment of DNA by joining DNA from two different sources. So initially, the DNA fragment was cut by restriction enzymes, creating the sticky end on both. Once another DNA fragment is cut with the similar restriction enzymes, you can then insert it because it will have corresponding unpaired nucleotides. This creates recombinant DNA. Another technique that is used is known as DNA amplification, and as the name suggests, you are amplifying or generating a large sample of DNA from a single gene or DNA fragment. There are two methods used in this process. One of these is through the use of cloning vectors. In short, you're using bacteria or a virus to produce many copies of a foreign DNA sequence. The foreign DNA is spliced into the DNA of either the virus or bacteria, and the bacteria and virus do their thing. They reproduce. As the bacteria reproduce, they are producing copies of the DNA that was inserted. This can then be harvested for use at a later date. Another method that is used is known as the polymerase chain reaction, or PCR. Here, it is an automated method of reproducing DNA by using enzymes such as DNA polymerase and primers. The steps are as follows. Initially, the DNA section of interest is placed in a solution of free nucleotides. Later, these nucleotides will be used to make copies of the DNA section. The DNA is then heated to open up the double helix. DNA polymerase is added, and DNA replication starts to occur. This cycle will be repeated many times, and the end result is lots of DNA sequence that is what you want to attain. To illustrate, the initial process is the DNA is chopped up into small sections to what you wish to find. Flankers, primers are flanked around the region of interest. It is broken up and the primers stick in. New DNA is synthesized in vitro. This process is repeated, one fragment becoming 2, becoming 4, becoming 8, 16, and so on. And this procedure can be automated using DNA polymerases from heat-loving or thermophilic organisms. Gel electrophoresis is a means by which DNA can be type sequenced. Here, using gel electrophoresis, fragments of DNA are separated according to their mass and electrical charge. On one end of the gel plate, a sample of DNA fragment is placed. The gel is then given an electrical current, causing it to become polarized on the other end. The fragments were negatively charged and they are attracted to the other end of the gel. The fragments move according to their mass. The smaller fragments move more quickly towards the opposite end of the gel. After a while, a characteristic pattern of bands emerge, and this is known as the DNA fingerprint of a person. Here you see the process illustrated. Initially, in an agarose gel mesh, DNA samples are placed into the gel. It is charged by placing an electric charge. The DNA starts moving towards the opposite end, towards the positive end. As it does, the smaller ones move quickest, the larger fragments stay back. From there, a dye, a staining procedure is used to actually cause it to glow, to actually reveal distinct bands. An example which you see here. As you can clearly see, this sample here on the left is very similar to this sample here on the right. This allows you to type and match DNA sequences. It's certainly very important in terms of forensic biology. And DNA sequencing is what can be accomplished as a result. DNA sequencing is the process of reading DNA fragments so as to determine the exact sequence of base pairs in a sample of DNA. On past public examinations, questions have been posed on this topic. I present one of these here for your consideration. What is the function of a restriction enzyme? Carry DNA to a new cell? That would not be the function of it. It does not function to transport DNA. Neither does it function to link together fragments of DNA or to make copies. Restriction enzymes cut and create sticky sites, the correct answer then being B. This question, as was presented on a past public examination, asks, along with restriction enzymes and gel electrophoresis, what is the minimum requirement necessary to perform a DNA fingerprint? Well, you would need a sample of DNA. So therefore, the correct answer would be either A or B. 2 would not be needed. Recombinant DNA is putting the DNA of another source in. If you were looking to perform a DNA fingerprint isolated to a specific person or organism, the correct answer would be A, one sample of DNA and the means by which to generate multiple copies, Pcr amplification. And this question as posed asks, how can the technique shown below in the diagram be used to analyze a sample of DNA? The first step in this question is for you to recognize that this diagram illustrates gel electrophoresis. Gel electrophoresis separates DNA fragments based on their size. The correct answer then would be C. Thank you for your patience. I hope you found this learning clip to be helpful.